Hi, this is Alex from devstories.eu. Scanpy, Single Cell Analysis, Episode 11 of 16. You have a perfectly annotated reference dataset and a brand new patient sample that you need to analyze. Normally, combining them means running batch correction, recalculating your principal components, and waiting for a massive joint UMAP to render from scratch. Data integration with Ingest offers a faster, non-destructive alternative. People often confuse Ingest with standard batch correction. Traditional batch correction tools take multiple datasets and calculate a brand new joint model. They alter the underlying representation of your reference data to force an alignment. Ingest does the opposite. It is an asymmetric projection. Your reference dataset acts as the absolute ground truth. The spatial model is locked in place, and the new data is simply pushed through it without altering the original model at all. Take a beautifully annotated peripheral blood mononuclear cell reference atlas. Every cluster is verified and labeled. You just received an unannotated, messy patient sample. You want those atlas labels applied to your new sample, and you want to plot the new cells in the exact same coordinate space as the reference. To make this work, the variables in your two datasets must align. This means both datasets need to share the exact same genes. In practice, you filter your new query dataset so its genes match the highly variable genes already identified in your reference atlas. Your reference dataset must be fully processed before you begin. It needs an existing principal component analysis, a calculated neighborhood graph, and a UMAP representation. It also holds the categorical labels you want to transfer, stored in its observation metadata. The execution is a single command. You call the ingest function, passing it your new query dataset, your annotated reference dataset, and the specific observation column you want to map, such as the cell type label. Here is the key insight. When you trigger the function, Ingest takes the expression profiles of your new cells and projects them mathematically into the existing principal component space of the reference atlas. It skips calculating a new global principal component analysis entirely. Once the query cells land in that shared spatial layout, the algorithm searches for nearest neighbors. It maps the query cells directly onto the pre-existing neighbor graph of the reference dataset. The heavy computational lifting of building a graph has already been done by the reference model. Because the new cells now have established neighbors in the reference data, two final transfers occur. First, the UMAP coordinates from the reference neighbors are assigned to the new cells. Second, the metadata labels, like your cell types, are copied over based on majority voting from those closest reference neighbors. The result is a query dataset carrying the exact UMAP layout and cell type annotations of your atlas. You can overlay the previously unannotated patient sample directly on top of your reference visualization, and the matching biological populations will drop cleanly into the established visual clusters. By projecting new cells onto an existing model, Ingest shifts your workflow from constantly rebuilding fragile global spaces to constructing one robust reference atlas and seamlessly flowing all future experiments through it. That is all for this one. Thanks for listening and keep building.